in the world of psychiatry. He's not only a psychiatrist, but a behavioural sleep medicine specialist and physician. And as you can see, he works at University College London Hospitals. Nick has a background of research in psychosis and circadian mechanisms and monitoring in psychosis, and is currently working, as I said, in the sleep medicine field, but also research actor, and has been very helpful in stimulating research in the world of circadian science, mostly for work as well, for example. So Nick, really looking forward to your talk. I hope it lives up to the excellence of the title. I'm sure it will, and I think it'll be a nice complement to what Anna's already talked about. So thanks a lot, Nick.
I'll try without the mic, but please put your hand up if you can't hear, and I'll use it. So really, thank you very much for having me. It's a real honour to be here. There are plenty of people here in the audience who'd probably be better qualified to be giving this talk. I made significant contributions to the field, but nonetheless it's an area of great interest for me. I spend a lot of time reading about circadian mental health, and obviously I see lots of patients with these problems. So if anything, what I want to really try and transmit and share with you is my enthusiasm and thoughts for how science, circadian biology can be intertwined and translated. All right, so some disclosures, none of which have any importance to this talk. So the temptation for a sort of talk like this is to really go for the modern, the cutting edge stuff, the GWAS studies, the machine modelling approaches, showing big heat maps. But I resisted that temptation because my feeling, having spent the past few years getting into grips with this field, is a really firm understanding of circadian biology and circadian science is essential. Whether you come from clinical, particularly those that come from a clinical background or working in psychiatry, I think we really have to understand circadian systems and the really rich and extraordinary research that's been done. So I'm going to spend the first quarter of an hour or so going through the top of this schema and talking about some of the landmark studies. It's certainly non-exhaustive, it's just a snapshot, but I want to try and create a narrative of really all the fascinating work that's been done, mainly in humans. And then the next half I'm going to talk about clinical side and how that's been translated thus far into psychiatry, particularly mood disorders. And
is that better?
Yes.
My voice doesn't carry so well. And yeah, what was interesting in doing this is you've got the top. The chronobiology is controlled, neat, tidy studies with unambiguous conclusions. And as is often the case, you look at the clinical studies, they're really mixed, heterogeneous samples, small studies. That's the nature of doing these studies. And the results are often messy and ambiguous, and that's one of the challenges we live with. Okay. So, starting sort of going back to the 60s. Going back before that, in evolutionary time, so we've evolved on the Earth, which has skins with a period of 24 hours, and all our, throughout our millions of years of evolution, we've lived under a 24-hour light-dark cycle. And that's had profound implications on our circadian, on our biology. We have clocks in us that anticipate the light-dark cycle and allow us to preempt and predict changes in the environment so that we can be active during the light phase, which is diurnal animals, and rest during the night phase. And that has certain evolutionary advantages. And within the past 100, 120 years, a blink of evolutionary time, we've subverted these natural rhythms, particularly by using manufactured light, developing manufactured light, which allows us to be active during the dark phase, and also living, for many of us, in urban environments, so we're covered and shielded from daylight. And this leads to a compelling hypothesis that these changes, particularly light changes, have really have profound implications on our lives, but also our mental health. And I think there's a lot of work still to be done to prove that hypothesis and to really make it secure, but I think it's a really fundamentally interesting one. So our story starts I'm starting here, it starts way before this. I've done lots of work in unicellular organisms, algae, etc. But Richter was working in Johns Hopkins. He was a scientist using mainly Norwegian rats. And it's just a nice example. of early chronobiology. So we have rats that tend to be active in the dark phase, and then the rats were blinded, and they ran wheels, and the real running was logged. And after blinding, the rest activity pattern of these rats gradually started advancing by a predictable amount every day, several minutes every day. So that suggests light plays a fundamental role in regulating a normal circadian rhythm. Then there was a hypothalamic lesion, which seemed to lead to this asynchronous, irregular rhythm. And interestingly, that's the kind of rhythm we see in people with advanced dementias. They really don't have sleep that's segregated into any one period. It just happens across the 24-hour period. And then another interesting finding from these series of experiments is that some animals had a different pattern. So these are 3 different animals. The 2 on the left had a gradual delaying pattern. The one on the right seemed to have a gradually advancing pattern. The animal on the right seemed to have a gradually delaying one. So that suggests there's individual differences in the period at which these animals free run. Then in humans, you're probably aware of the bunker experiment. So this is Jürgen Aschoff and Rutger Weaver, who did some seminal work in a bunker in Bavaria, and healthy human subjects spent several weeks in total in a dark bunker with no reference to external time and controlled light. They could self-select light, but the illuminance was controlled. And what they found, also while measuring core body temperature, is that people's sleep timing tended to drift, so there was this gradual phase delay, and their core body temperature also tended to drift. With slightly higher illuminance, it still drifted, but with a slightly lower period of 24.4 hours. But essentially, the conclusions at that time were the human intrinsic period was around 25 hours. They also noticed another phenomenon of what's called internal or spontaneous desynchrony. So the period of the pacemaker, or the core body temperature, had a certain period that dissociated from the period of the sleep-wake cycle. So the 2 drifted apart and oscillated at different periods. And that's something we certainly see clinically in patients with psychiatric disorders. So that's some of the groundwork. And then in the early 80s, there's some genetic work done. The period gene was was identified and cloned. And then in the late 80s and 90s, the translational feedback loop was proposed and formalized. So this is where the transcription factors COP and BMAL initiate expression of clock genes CRY and PER. These heterodimers go into the cytoplasm and then inhibit their own transcription. And this is a continuous loop with a period of around 24 hours. So we can see PER CRY levels go up and go down over 24 hours. And since then, we know there are lots of polymorphisms, variants in these clock genes, both clock, PER, and CRY, for example, and there are probably many more that seem to have bearing on people's individual circadian rhythms and periods. And also, we now, there's been subsequent work done, and I know some people here involved in this work, where there seems to be organ-specific expression. So really a large number of our proteins that are expressed in our body have some circadian rhythmicity, probably controlled by core clock genes. And in humans, probably around 50% of our transcriptome or gene expression is rhythmic, with substantial organ specificity and time of day specificity. So moving on to the late 90s, this is work from the Seisler group in Harvard, and some really seminal experiments using forced desynchrony protocols, so very controlled experiments. Again, in healthy individuals. And this ascertained the human intrinsic period. The average intrinsic period is around 24.2 hours. So most people are above that. The majority of us have slightly longer circadian periods. A few have shorter circadian periods. And that's interesting. If you have a shorter period, you're more likely to phase advance over time. You need evening light to bring you back into entrainment with a light-dark cycle. If you have a longer intrinsic period, morning light brings you back in. And we have these concepts of period, phase, and amplitude of our circadian clock. And then, going back a bit to the early 80s, this really fundamental concept, which I use every day with my patients, of the 2-process model. So this is the constant dialogue and exchange between homeostatic sleep and circadian mechanism. So there's process S, which is a homeostatic kind of hourglass mechanism. It builds up with wakefulness and is paid back and dissipated during sleep. But our alertness through our waking day usually stays pretty stable. We might have a lull at around 3 p.m, but otherwise we maintain stable alertness despite this rising homeostatic sleep pressure. And the reason for that is the circadian system. It produces a continuous and growing wake-promoting signal through the day, and that suddenly drops off over a cliff at just before our habitual sleep time, and we fall asleep because you have unopposed homeostatic sleep pressure. So they, in health, need to be balanced and synchronized in time. If one slips out of sync, we may develop sleep problems and also psychiatric problems as well. Then we also have this thing called the wake maintenance zone, which is really interesting. It's actually really difficult to initiate sleep during that period. It's also the forbidden zone for sleep. And at the end of that period, there's what's called the sleep gate, which is when sleep can happen. And again, that's really important when thinking clinically. So I'm sure a lot of people appreciate this. I just want to really emphasize that these core principles are important, I think, in any future research we do. Okay.
So next.
We move on to the really amazing work that's been done around light and the human circadian system. So Anna was one of the investigators working on the phase response curve, and this is a formalization of the way in which light shifts our body clock and resets our body clock. So our circadian nadir is around 4 in the morning if we're normally entrained. That's when our core body temperature is lowest and our sleep propensity is highest. Light before that time delays our clock and pushes us later. Light after that time pushes us earlier. And probably around 3 to 4 hours before is when we have the strongest phase delaying effect, and vice versa for phase advance. And it's also since been known in other experiments that melatonin has a roughly converse, but less powerful phase response curve that's in opposition to that of light. So melatonin in the evening tends to advance the clock. And subsequent experiments have really shown that the human circadian system is exquisitely sensitive to light. So this is another landmark experiment by Jamie Zeitzer. And you see here, the more light someone has, the higher the illuminance, the greater their shift in their melatonin phase. So the more light shifts their clock earlier or later. And this curve saturates at about 10,000 lux, which is the intensity of sunrise or sunset broadly. But we still get half that phase shifting effect with 100 lux of illuminance, and that's the amount of light in a dim room light in the evening. So that amount of light still has a significant phase shifting effect. I think that's really important to bear in mind, to think about. And there's another important study in the late 2010s that it demonstrated significant individual variability in sensitivity to light. So there's a 50-fold difference here between those people in blue there with a very high sensitivity light. They're shifted, 50% of the melatonin suppression is suppressed at 10 lux, which is extremely dim light. And the low sensitivity people need up to 1,000 lux in order to have the same phase shifting effect. So I think there's a real awareness of the importance of this individual sensitivity. And then this is a nice study that I always like to reference, and this is a real world study of a so-called camping study. So a group of 8 people spent a week living in urban environments. They had light measurement, DILMO, and rest activity monitoring. And what you see when they live in the urban environment, in the light grey, their light levels are a lot lower, and they have this kind of shoulder of light in the evening, which isn't seen in the subsequent week when they're out camping in the Colorado mountains. So the light environment is profoundly different, and that has a real bearing on our sleep and our melatonin rhythms. So urban's at the top with electrical lighting, natural lighting below. In natural light, where you don't have that evening light, the sleep period is constrained, such that sleep offset happens quite close to sunrise, and also the melatonin offset, the circadian night, also ends at that time. In urban environments, our melatonin offsets a lot later, and happens a lot later and a lot after sunrise as well. So our light environments are really fundamental to our circadian biology. We have increasingly understood also that there's another sort of non-visual light pathway. And we know about the pathway to the SCN in the hypothalamus, but also light has effects on far-reaching parts of the brain, including the cortex, nucleus accumbens, so parts of the brain involved in emotion, amygdala, habenula, nuclear, etc. So light not only subserves visual function, it not only shifts the clock, but it also may affect mood, alertness, cognition through other pathways. Okay, so we're getting on towards the clinical sort of side of the talk now. So I think another fundamental question is, what is circadian disruption and misalignment? And what is circadian health? So circadian health, and I'm going to point these little things are going to oscillate, so this is a demonstration of what we might call circadian health. So there's a rhythmic environment in the world around us, and our internal clocks and our SCN and our periphery are all in alignment and oscillating with the same period and at the same time. And here's a model for what might be going on in one type of circadian disruption, so misalignment. Our central clock and our peripheral clocks might be out of alignment with the day-night cycle. Another configuration might be this. So our central clock might be aligned with the external world, but our peripheral clocks are out of sync with our central clock. So that's called internal desynchrony. And that, for example, might happen when you come off a shift. You're doing a night shift, and then you're eating at a different time. So your body clock's been trained to the time you're eating, but your central clock is doing something else. And that might cause pathology of its own. Another configuration might be like this. Your sleep and wake is out of sync with your body clocks and or the day-night cycle. So there are lots of complex ways in which misalignment and loss of coherence of oscillators might arise. Another important facet that I think we need to pay more attention to is amplitude. So circadian amplitude is the strength of the nadir and the peak of our circadian rhythms. And generally in health we have high amplitude rhythms. And in disease they might be lower amplitude, and that might also occur from things being in antiphase. So amplitude might also be a function of oscillations occurring out of phase. Okay, so how are we doing time-wise? Plenty of time. So, moving on to chronopsychiatry. This is a couple of figures from Richter's book, and it's long been noticed that in psychiatric illnesses they can follow rhythmic patterns. So in the first example on the left, we have someone with a mood disorder that seems to have daily sort of 24-hour cycles of euthymic mood interspersed with fear, low mood, and sleep seems to oscillate as well on a daily basis. Craiglin, who was working in the, I think, 20s and 30s, also noted similar patterns in people with manic depressive illness, with annual cycles of mania and or low mood. And Craiglin also noticed, this is a quote, that in these cases moodiness sets in at autumn, seems to pass in spring when the sap shoots in the trees, to excitement corresponding in a certain sense to the emotional changes which come over even healthy individuals at the changes of the seasons. So I think what he's talking to there is really a description of seasonal depression, so worse low mood in the autumn and maybe elevated, buoyant mood as spring comes. And also there's an example there of someone who's having twice yearly cycles. So we have all kinds of temporal rhythms that occur over different temporal timescales. Then we've heard a bit about Norm Rosenthal and Tom Ware, and who Anna knows, has worked with closely, and they really coined this concept of seasonal depression. So this is people who develop depression over winter months, and the depression is somewhat atypical. So Mood is negative, but also appetite can be increased, which in most types of depression we don't see. People tend to lose their appetite. There might be carbohydrate craving and increased weight. And also people tend to become hypersomnolent in this variant of depression. So in classical depression, people develop an insomnia type phenotype. So there seems to be this unique mood phenotype called seasonal affective disorder. And interestingly, this group seems to respond to light. So you give them bright light and extend their photoperiod, and the mood seems to improve. And so I had the pleasure of meeting Jacob, and I know Ian's here as well, so their group have been working on this circadian depression phenotype. And as you can see there's lots of overlap in their description of circadian disruption with that of the SAD. So it seems that bipolar depression, seasonal affective disorder, and this atypical depression phenotype really share common features. So an extension of that being there was a subgroup of people with depression where the depression is underpinned by circadian mechanisms. And this is in contrast to sort of, let's call it classical depression. We have a control, this is a hypnogram, so we put electrodes on the brain and measure the brain waves during sleep. And we see here, in a normal control, REM is mainly at the end of the night, slow wave sleeps at the beginning. In depression, you tend to see a bit of a change. You see REM happening earlier in the sleep episode, so the REM onset latency is shortened, and you have insomnia, so fragmentation of sleep both at the beginning and end of the night. So this picture actually is interestingly not seen so much in the seasonal affective group, which is interesting. So there might be changes in the ultradian structure of sleep as well. Going back to light seems to be really important. This is an interesting meta-analysis that was done in the 90s, where the risk of developing SAD was estimated according to latitude, so average photoperiod for a given latitude. And it seemed to follow this curvilinear relationship, where the further north you were and the shorter your average photoperiod, the more you were likely to develop SAD. Again, making quite a strong link between light exposure and mood. I think Greg Murray did a study in Australia that didn't show this relationship, but it's quite compelling. And since there have been many other studies, for example, those using UK BioLank data. This study used objective data from a watch from the AX3 device, and concluded that people who were exposed to more light at night had increased risk of all kinds of adverse mental health outcomes, as well as physical, other studies have shown. And independently of that, brighter light during the day was associated with decreased risk of these mental health disorders. A huge confound in this study, however, is it's cross-sectional, and there are issues around reverse causality, of course. So, where are we at the moment? So this is a healthy control, and blue is the DILMO, the salivary melatonin is starting to rise, and yellow is the circadian nadir of core body temperature. And we have something called the phase angle difference, which I'm sure many of you are familiar with, and this is the time difference, the interval between the onset of DILMO and a behaviour, and often the midpoint of sleep is used as the pad. So we all, let's say we're someone with a slightly longer circadian period than 24 hours, we have light in the morning and that shifts us back and entrains us to the 24-hour cycle. Someone with circadian depression, they may have, for whatever reason, they might have a longer circadian period, altered sensitivity to light, their circadian phase is delayed. So their DILMO is happening later in clock time, the core body temperature is later, with respect to their sleep timing as well. Some people may also have a delayed sleep phase in addition, which sort of complicates the picture a bit. So there's pretty secure evidence in depression, in non-seasonal depression, that there's a group with this phase delay. There's several studies that have shown this. In seasonal affective disorder, actually, it's not as robust. So we see some people with a shorter phase angle and a delayed circadian phase, but some people with phase advance. So one example of this is our Louis study, which showed there's a group here who are delayed, so their phase angle difference is longer than 6 hours, which is considered an average. Some people, however, are advanced. So the SAD literature is less secure in terms of pathophysiology, which I think is an interesting thing to think about. And Aloui proposes that actually it's what he calls ipsative differences, which is within person shifts that's more important than absolute shifts in sleep timing, in circadian timing. So this is a sort of cartoon of an intervention in circadian depression. We use morning light and or evening melatonin to pull the clock forward, advance it. And again, in circadian depression, or in depression, or in non-seasonal depression, that intervention is effective, both in advancing sleep timing or sleep preference, i.e. chronotype, and that leads to an improvement in mood. So light therapy, morning light therapy in particular, some studies have shown melatonin as well, seems to lead to improvements. And those with delayed sleep seem to have worse depression and benefit more from these interventions. A drawback, however, is a lot of these studies have only measured proxy circadian markers. So they've measured rest activity rhythms or chronotype and haven't looked at endogenous markers of circadian phase. So it's not really, hasn't really been shown that advancing circadian phase leads to these improvements. As yet, there's still further work to be done. Seasonal affective disorder, I think we can have a big debate about this, but again, the evidence overall suggests that light therapy improves outcomes, improves depression. But I'd say the evidence is quite mixed, an issue being relatively small studies, small study samples, problems with controlling the interventions. So I'm going to move on and look a bit at the bipolar literature, and there's a huge corpus of work done in bipolar literature, bipolar disorder, around the neurobiology and the involvement of circadian rhythms. So there's a lot, too much to cover in this talk. I'm just going to touch on a little bit. So firstly, the International Society of Bipolar Disorders had done 3 really great reviews about 3-4 years ago. And similar to unipolar depression, they conclude that bright light therapy has a sufficient evidence base to recommend it in bipolar depression. Sleep deprivation, as Anna talked about this morning, also is recommended, albeit with slightly less strength of evidence. Dark therapy in mania, and interpersonal social rhythm therapy is a good evidence base in treating bipolar depression, but also preventing relapse and maintaining remission. Vincent spoke about lithium really compellingly a moment ago. There's a really interesting work around this as well. And this is work by Michael McCarthy and colleagues in San Diego. So this is taking a group of people who have responded with bipolar disorder, some of whom have responded to lithium, others who haven't. And you can culture their cells, either through skin fibroblasts or pluripotent stem cells, and mark their circadian expression with luciferase and look at the period and amplitude of their circadian rhythms. And the groups segregated into, well, the lithium responsive people tended to have shorter circadian periods and more morningness chronotype. Those lithium non-responsive people tended to have longer circadian periods that wouldn't shorten with lithium ex vivo. So again, really potentially interesting personalised medicine approaches here, where we might be able to target lithium therapies to certain populations, or we might be able to find other medications that target the circadian rhythm without having the side effects that lithium does. Lithium can be a very effective drug, but obviously has lots of side effects. In a similar vein, there's been this sort of quite heroic study. I think it's a study with lots of issues, but nonetheless it's tried to measure circadian function, amplitude and phase at different affective states. So when people were manic, their cortisol level and their buccal cells were used to look at their circadian phase, and they were phase advanced, up to 7 hours phase advanced during mania, and when they recovered to euthymic phase, they had phase delayed. Conversely, in depression, people had phase delay during a depressive episode which advanced during recovery. It's very hard to do these studies, but potentially interesting to look, interesting to think about. And there's a lot of other work that's used actigraphy, and there's a poster out there today that has shown flattened amplitude and generally phase delay and tendency to phase delay in depression as well using actigraphy, and those with more disturbed rhythms have poor outcomes, including more frequent relapse. Okay, I'm going to just skip through this. That's some schizophrenia work. So, where next? So I'm going to offer just a few high-level points. There are many more. These are just things that kind of came to my mind when I was putting this talk together. So the first is the importance of defining circadian dysfunction, and we're still not there really. It's sort of, there's an intuitive sense of what circadian misalignment is and circadian reduced amplitude is, but we haven't really operationalized it or formed a consensus on that. It might be something that a Delphi group approach could be helpful with. But what constitutes abnormal phase relationship depends on age. It changes across the lifespan, and it differs by sex and chronotype, so that needs to be incorporated into these definitions. And temporal plasticity of the circadian system is adaptive and maladaptive at different times. So, when we have jet lag, we need to reset our rhythms and put them back into phase. That's an adaptive change, or changing to day length, for example. So that needs to be factored in. We need to understand the circadian system in context. So everything I've spoken about is very circadian, but circadian disruption might cause disease, not simply by the fact that it's circadian disruption, but by virtue of its impact on sleep and lots of other associated functions or processes. So the circadian system is dynamic, it's embedded and interconnected with everything else. So we need to understand and carve out what is circadian, but we also need to understand everything that it sits in. So a simple example, which many of you will be aware of, is an adolescent here with phase delay. They have circadian phase delay for reasons that we've gone through. Their sleep phase is delayed, but they need to get up at a fixed time in order to meet social commitments such as going to school. So their sleep duration, first of all, is truncated. They're experiencing sleep loss. They wake up with more sleep inertia, might have trouble focusing at school, might having trouble social interactions. That might lead to lower mood. So these are simple and well-rehearsed paths through which circadian disruption might lead to low mood. There might also be another process, might be REM sleep. So we know REM occurs in the second half of the night. If you truncate sleep, you might prematurely reduce the amount of REM. We know REM is involved in emotional regulation, for example. So there are all kinds of pathways to think about. We generally think of the circadian system as sort of the top of a hierarchy, so it governs so many other brain and body functions. But also understanding there to be a reciprocal interaction between sleep and the circadian system. So sleep, when we close our eyes, that gates our light-dark exposure. And similarly, there's evidence to show that sleep deprivation changes the way in which our circadian system shifts and makes it less sensitive. So, yeah, the circadian systems interact in a reciprocal with other body systems. And Dirk Jan and Paul Franken, I think, wrote this really nice review last year on this. So it's worth reading. We need to measure sleep and circadian function together. So that's quite grammatically ambiguous. We need to measure circadian and sleep together, but also together as a group of people. We need to do this. So a lot of the studies have used these sort of composite phenotypes. So sleep timing using rest activity rhythms, actigraphy, measured chronotype, and that there's all really important measures, but we need to understand what they're measuring. They're certainly not just measuring circadian function. Chronotype is affected by homeostatic sleep, pressure, or the dynamics of the homeostatic sleep system. It's affected by mood, for example, personality type. So it's really confounded. So I think future work, for example, will involve trying to parse out and understand and validate something like accelerometry against endogenous circadian markers. And that will then open up and allow us to interpret, for example, biobank studies with more precision. And it's all very well saying, there's a 60% correlation or 80% correlation between our chronotype and our circadian phase, but there'll be outliers, and it's those outliers are important. We're talking about people with delayed sleep phase or very advanced phase. And for them, something like the phase of rest activity rhythm doesn't correlate so well. So there's lots of further work to be done there. There's also the promise of using more single point of access tests, blood-borne markers, blood-based markers, white cells, fibroblasts, for measuring circadian phase and amplitude and period with more ease. And those are in development, and I think it might be quite an exciting research tool to use, and allows for personalised precision medicine. We also need to be measuring light and doing these together with those other measures we've spoken about. There's really a lot of work's been done. Manuel Spitzham, for example, for one, has done a lot of work in developing a checklist for reporting light in melanoptic alpha light. So measuring and reporting light in circadian context. And this is a device by Sean Kane's group, which is a sort of clip-on device that does that as well, and you can wear it on a lapel. There are again standards around measuring light in these circadian experiments that I think we really need to attend to. So this is an example for one of my patients in my studies. I had a year of accelerometry. So this is interesting, and it showed this spontaneous desynchronization over winter months. But how much more interesting would it be if we had light exposure during this time? At the moment it's only a hypothesis that this person had less light over winter and therefore desynchronized. How much more interesting would it be if we also had circadian phase markers for this person? So did their circadian phase remain entrained during that desynchronization, or did it slip along with their sleep-wake rhythm? The interesting thing in this patient is you can also see the next winter she did the same thing. This is a patient with psychotic illness. So there is, I think, there's a lot of interesting work that can be done through longitudinal measurement of simple things like rest activity, but also adding light, also adding endogenous markers into that. We need to attend to individual differences, and people are doing this work. They're looking at light sensitivity, polymorphisms in circadian genes, for example, that might render us sensitive or less sensitive to light. And therefore, we can tailor, for example, our diet, so how much light we need in order to remain entrained. But part of this is also sleep. There are inter-individual differences in sleep, homeostatic sleep as well, that we need to look at together with the circadian side of things. And final set of slides, this is about acknowledging phenotypic complexity and comorbidity. So the psychiatrists among us will be very used to this. We know that in many of our disorders there's significant overlap. Risk genes for bipolar disorder and schizophrenia overlap. Psychopathology overlaps, similarly anxiety and depression. So there's this really kind of messy clusters of disorders often. Similarly in the sleep world, right, we have insomnia, and that's the most commonly presenting sleep phenotype, but within that is intermixed, or is likely to be intermixed, other sleep phenotypes. So especially circadian rhythm disorders, we know that, I'll show you a slide next that suggests a proportion of people with insomnia have phase delay. Similarly, hypersomnia might also be the case. That people that are phase delayed tend to have longer sleep periods and can have hypersomnia phenotype as well. So this is a study from various people, but Australian group, showing people with insomnia. So the main panel shows people with insomnia who are appropriately phased, so their DILMO is occurring over 1 hour before their preferred sleep onset time. There are a group of insomnia patients though where the DILMO is very at or after their sleep onset time, so they're abnormally phased. So these might be a delayed circadian type, and they're presenting with insomnia because they're trying to go to bed at their sleep forbidden zone of sleep, and they're not able to sleep, and they have an insomnia phenotype. So trying to parse out these sleep phenotypes as well as psychiatric phenotypes is going to be important. Alison Harvey wrote this brilliant review in 22. I generally find most of what she writes is worth reading. And this is part of her Trans-C trial, so it's a CBTR type intervention for SMI. And the vast majority of people in this cohort of people with severe mental and enduring mental illness had 2 or more sleep disorders. One of those might be sleep threshold, but essentially comorbidity and sleep disorder is the rule. So yes, we need to look at circadian disorders and dysfunction, but we need to think about how that expresses itself in other sleep disorders, including insomnia, including hypersomnia. And Sarah Reed at Oxford had made a similar finding in a psychosis group. So, insomnia was certainly prevalent, as was nightmare disorder, hallucinations, restless legs, circadian rhythm disorders. She concluded in this paper that each patient had 3.3 sleep disorders on average. So, worth thinking about. So I'm going to end with those conclusions there. And invite any discussion that arises. So thanks for your attention.
Thanks very much. Thank you.